Hello, fellow neuroimagers. Today we are going to cover the fourth lab in the series, or Lab 04. There weren't a lot of common gotchas in this lab, so I felt like it was pretty straightforward. So to start out here, we're going to open up the terminal, cd to our Iowa Labs, your username, and I'll also open up my Jupyter notebook here. And I'll get this screen lengthened if it will let me. Maybe it won't. There we go. Kind of 04 lab. Jupyter notebook. Okay. So I'm just going to run the top here so that we have that ready to go. And I already have the files in place. So we're going to scroll down here to the exercises. Okay, I already ran epireg because that command takes about, well, it can take 15 to 20 minutes. So I don't think that you guys just want to sit here and watch me run a command for 20 minutes. So I already have that pre-baked. I'll walk through a little bit of the command since it might provide some details, but the only thing of note that I can say here that makes epireg different from other FSL commands is that we have like this double dash notation and then an equal sign. So this is just a convention within FSL in which if you see 1 and 2 dashes before an option or flag, then in order to set that variable, you have to have an equal sign after. Say if I got rid of this equal sign and tried to run it, it would not let me run. So that is some little idiosyncrasy about FSL just to be aware of, that when you see the double dashes, that typically means that you need to put an equal sign after the flag. So now we can comment on whether we think BBR did a better job. And then just to be completely explicit, epireg computes the brain-based registration, so epireg computes BB, the brain-based registration. So in order to check this out, we will open up FSL View with our corrected images. So let's see if I remember. Oop, actually I need to make it a bash shell. Or I guess, oh yeah, never mind. I need to actually make a code block. That'll be code, and then I'll make that bash, and I'll do FSL View with that one, func, that one, and my T1. And then we will be able to compare all the registrations. Okay. Move that up. I'll move this one up. Okay. Change the opacity so we can see through a little bit. So we can see as I just flip between the two images, looks like there's pretty good alignment. with the BBR method, and if I switch that out with the 6DOF, and we'll just flip back and forth a couple times here. Honestly, that one looks pretty good too. Just switch one more time. So the only difference between the two from my end is that I didn't use the skull-stretch version of the example func when I computed func to T1 BBR. If I did, then these two may have been a little bit more comparable. But just to get another quantitative view, we can also check out the mat files. So I'll insert another code block here. And we'll just keep using bash, and then I will cat that mat file. And then I'll insert one more cell below. And I'll do the same. Cat func bbr.mat. Alright, so if I do this one, let's look at this one. So now we can see exactly what the differences are. So if you recall, along the diagonal is the scaling factors for the x, y, and z axes accordingly. So we see that there is not a great difference between the two methods in terms of scaling, so they both approximated that pretty well. And for rotations and some translations, those are a little bit harder to interpret, but again, a good place to read all about those affine transformations and how they break down into their component parts, even though they might not be necessarily interpretable, but you can look at how they break down by rotation. And this one's the scaling. All right. And the other methods to break down the affine transformation to its component parts. So I'm just going to stick to the very easily interpretable ones, such as the origin. So we can see that this is where they came up with a little bit of a difference, where is the 6 degrees of freedom, we have 12, 13, 60 as our transformation from the bold's origin, or the example func's origin, to the T1 origin. And with the BBR, we have 10, 16, 66 moving to the origin, to the T1 origin. So that is at least that is the most noticeable difference that we can see between these two transformations. Just exactly where they centered it. So BBR should theoretically do a better job, especially if there's good gray-white matter contrast, as we mentioned in class today. If your data don't have a very good gray-white matter contrast, you can get some pretty horrible results. It should be pretty noticeable with smearing of the data essentially across. For ours, it was the x-axis. It was just smeared. The brain was entirely smeared across that, so it looked very bad. So with that, we'll move on to question number 2. So we'll extract a different region of interest. So our transform set, we will use the BBR, so just that we have extra practice with that. So we'll do a concatenation of all the warps here. Eliminate that. Okay. Then we still want to maintain a solid name, so the reference is still going to be that. Our premat is now going to be the funct2t1. bbr.mat, I believe that's what we named the output. And then still the warp is going to be the same from the T1 to MNI since we've already evaluated that. Then we'll call this one BBR warp. So again, we see that double dash equal sign notation for this command. And now I'll run it. Now the next step after we concatenate the warps, we need to invert them, or invert the invert the func to MNI BBR warp. So again, we have that warp, and now I need to convert this one. I guess let's see, how do we apply that warp? Warp MNI to T1. Alright, let's see if we can actually do a one-shot from MNI to from MNI to func, just with one warp. So we'll see how successful I am at that. Make sure I copied the right thing. Okay. I'll see. Alright. So we got the T1, but we're actually going to use this warp right here. So I'll copy that one. And the output will be MNI to func BBR warp, and my reference image will be the example func that we have. Alright, and it wouldn't matter if I chose the brain. All the reference image is doing is deciding how the image should be resliced, as in the voxel sizes of the final image, which do not change between the Skullstrut version and the non-Skullstrut version. So I will begin to run this invalid. Oh yes, of course I forgot to make it a bash shell. Bash, there we go. So now that we have that warp inverted, we can take an arbitrary value okay, so we can insert another. I'll take an arbitrary value, which is 9. 9. And I thought that was the amygdala, but let's call it, leave it a surprise and call it my ROI and MNI. .gz. Okay, so again, I forget to call it a bash shell. I'll do that. Okay. And now the next step should be just applying the warp. So we have that function defined here. Let's Control C. Insert another cell below. Make sure it's a bash shell. Then let's do the apply warp command. So the input will be my ROI, MNI.gz. You can either leave or keep the extension on. The reference is still, can either be the example func or the example func brain. The output will be my ROI mask func, and then the warp we're going to use is the one that we defined here, so MNI to func. Func BBR warp, and since we've combined the mat, the matrix file, we don't need to include that here. When I say I combined it, I mean that we applied it here. So theoretically, we'll see if this invorp actually inverts the matrix as well. So we'll run that, the apply warp, it finished instantaneously. And I think we are ready to check it out. So let's just do interactive check here to see if it looks horrible. Cell below, we'll make another bash shell. And instead of the albutamen, this one is the my ROI mask func, and let's give it a go. So let's make this a different color so that we'll be able to see it. Okay, so based off of this, I'm going to guess the left hippocampus. So let's see if my guess was right. I'll use another FSLView command. Insert cell below, FSLView. Should I figure out what the name of the 04 lab? The name of the atlas. Okay, so it's called the Harvard Oxford. This guy. So take that. FSLView. Let's use this one again. And we'll make this a bit better looking with, let's call it random rainbow. number 9. Okay, so tools, toolbars, Atlas tools. That's the left hippocampus, number 9. Alright, so we have confirmed that that is what it is. So finally we're going to use this tool, the Nylearn plotting tool, to actually give us a nice printable, shareable figure. So that can still be the example func. This will now become my ROI mask func.nai.gz. Then plot it. We'll add a few more cut chords. Oop. I think I changed that to markdown on accident. Change that back to code. Okay. And then we will see what pops out. I should change the title here too. Or at least it should show this pretty quickly. Oh yes, it was waiting on my FSL view command to finish. There we go. And then here they actually display the left on the left side, so we can see that the hippocampus is on the left. I think that it's matched up correctly. I might check a little bit later and let you know in the comments of the video or on the Slack group to confirm that the hippocampus is in the correct place. But until then, I believe that is everything that I wanted to show you for this lab. And all I should have done is change the title there to something different so that it would actually be a presentable figure that we could save as a PNG and share with our collaborators or put in a paper. Yeah, everyone, great job on this lab. If you weren't clear on anything, hopefully this video helps clear it up. And that will be it. Thank you all for listening.